It's a pleasure to follow up, Leon. It's all the pet projects, especially CSOs, which I don't have to talk about today. And there's a few other slides that I don't have to talk about as well, because we overlap really, really well. So I'll do my best to entertain you. I'm going to be talking about wastewater epidemiology, but the biology side of things primarily. So that's just a walk through what I'm going to do. So it all, I mean, it doesn't actually start here, but it's almost like the heart of where wastewater surveillance kind of feels like it started. So sort of down the street, metaphorically, there was one of many epidemics of cholera. They didn't know it was cholera at the time. The chief medical officer role was created a few years before the 1854 epidemic, who actually just came to visit last week to talk about CSOs and the health threat that it poses. So he's very proud of the fact that he's maintaining the tradition of his role. And that's basically that sewers were a huge health problem. They obviously still are, but possibly not as bad as 500 deaths in 10 days, which was the epidemic at the time. And so this is a map that Edmund Cooper was credited with, which basically outlined where the homes are that had deaths and then how many deaths per home. And, but didn't know necessarily, the short version is, didn't really quite know what to do with it until John Snow came around, came up with the idea that maybe it was sewage, and it was subsequently demonstrated to be sewage. So sewage is playing a role in human health. It was obviously doing it for quite a lot longer than since 1854, but it was the link between human health and exposure to sewage was very clearly made from that point forward. Moving forward, I think 85 years, I put it down there so I didn't have to do math on the fly. So this is back in America. And there is a team up in Yale who were doing some research on polio, and they acknowledged that others were finding polio in the stools of people afflicted with polio. And they thought, well, that's interesting. I wonder if there was an epidemic, we could find it in the sewage, which is, you know, it seems an obvious thing to ask, but it wasn't an obvious thing to ask back then. And they so had a go. And back then, when you have a go, it's a science paper. So it was maybe a little bit easier back then. So this was a 3-month experiment done sort of opportunistically. And they looked for the virus, and they found it. And there was a lot more virus in that sewer than in sewers where the epidemic wasn't present. And so truly, wastewater epidemiology is born here. Unfortunately, well, they were constrained by their time, and so at this time they were having to culture the virus, and so there was no molecular approaches naturally back then. However, did I put the, yeah, 49 years later, the molecular approaches are getting a bit more routine, and so a further outbreak of polio was demonstrated to be occurring in Israel. And this was sort of the birth of molecular epidemiology for polio outbreak tracking. And so what they did is they went to the sewage, they extracted the nucleic acids, sequenced it, started to do it in enough places to see that mutations were starting to accumulate in some locations, and they weren't present in other locations. And then started to recognize, well, obviously they would have known this, is that the longer it's basically being passed from human to human, the more it accumulates mutations. And so they could basically track back where the origin of the epidemic had occurred. And so there you go, molecular epidemiology using wastewater was born. Now, this is coming down from the grand heights of all that work. This is work that I did back in the the swine flu pandemic of 2009. So all scientists like opportunities such as the pandemic to sort of test out ideas, and this one was no different. So what we did is we went to the wastewater and rivers, and we sampled them. This particular study was based on just because we were constrained at the time, as you always are, we basically went to 2 different sewage works and repeatedly sampled them and tried to understand the relationship of the metabolite. So you take Tamiflu, it's a prodrug, goes in your body, it changes it, basically metabolizes it to the active antiviral. That metabolism is proof-ish that the drug was actually consumed. So it's as good as you're going to get, really. So if we're looking for the metabolite, it basically is a confirmation that it wasn't just dumped down the toilet. And so we looked at the prescribing practices, and so there's 2 pieces of data for prescribing. One is the HPA, but that data wasn't a particularly good data set. They basically undercounted the amount of people who were being prescribed. And then the acronym is NPFS, is effectively the service that was responsible for prescribing or effectively disseminating the Tamiflu to the population. So they actually had the data, whereas HPA had more about hospital visits and things like that. So the NPFS is basically you can generate an estimate of what you could what you predict to find in wastewater if 100% of the population consumes it versus lower populations. And so if you look at the load of the drug in the wastewater, it matches up with quite a low estimate for compliance. So I think it's roughly 45 to 55% of the population was consuming the drug despite having been having collected the drug from the chemist. So this is really about the behavior of the public in a time of crisis. Now, had this study been done, let's say in June, July, when the pandemic was quite new and everyone was a bit panicked about it, I bet you that the load would have been at the high end, so it would have been that compliance was quite good. But we did the study tail end of October, so everyone had some time to sort of marinate in all of the media and in their own experience of how serious of a pandemic this actually was. And by then, they realized actually it's not much worse than a normal pandemic, seasonal flu, and they didn't take any drugs for seasonal flu, so they're probably not going to take the drug now. And so the compliance rate went way down. Turns out compliance rates are really quite low for a lot of drugs. The only drugs that they're not low for are chemotherapy drugs, which you can imagine why. So during the same period of time, we recognized that Tamiflu is not the only drug that's being used during the influenza pandemic, and that antibiotics are substantially increased. Their use is substantially increased during a pandemic, or at least the influenza pandemic, because of secondary bacterial infections. So what we did is this is actually quite a huge piece of work. And so we use a global model for influenza pandemic spread, and then effectively just did the Thames catchment, so where you're sort of sitting at about there. And all the dots refer to wastewater treatment plants of a certain size and above, so it doesn't include all the really small ones, which there are quite a lot of them. And so we know what kind of prescribing of the population is at every location. You know that they're going to poo and pee, and it's all going to go to one of those wastewater treatment plants. And then because we know the concentration that this will produce, assuming a degree of metabolism that's going to go on and loss during transit, and also loss in the wastewater treatment plant itself, we can predict what the concentration in the wastewater treatment plant is going to be, and we can predict what it's going to be in the river. And so the color of the dots are indicative of the toxicity. So basically, this is an antibiotic model of toxicity. This came before a lot of work that started to look at resistance. So we're looking at the death of bacteria, not actually selection for resistance, which would have been 10 to 100 fold lower in concentration. So everything would have been red. So this is just effectively death of bacteria. And so depending on the size of the pandemic, so you can see the R0 values, the pandemic itself was roughly the one on the left. So there's not a huge increase in the concentration relative to what we were doing already, because we use a fantastic amount of antibiotics all the time. And so a small, roughly 3% increase was not going to change the game too much. But once you start ramping up the pandemic, the response using antibiotics will likely result in the loss of the functioning of sewage works. And then the downstream rivers will microbiologically either generate fantastic amount of resistance, or they'll start to lose their function. Right, so all of this is stuff I can skip over, but I just wanted to say that this plays a really important role in almost like anchoring the wastewater epidemiology field, because it's been around for so long. It's generating really, really valuable data and award-generating data. So this is the anchor to wastewater epidemiology. Now, this is probably the area where there's the most opportunity, because as you've just been told, the instrumentation can generate billions of data points. And no one's analyzing a billion data points. But we could, if you generate the algorithms to analyze it. And so biomarkers effectively talk about these, it's a placeholder word for everything that comes out of you when you into the toilet and then gets flushed. And that could, speaking about the people this morning, air pollution will generate biomarkers. If you are in this city inhaling the air, you will have biomarkers that are unique to high pollution cities. And if you go to a low pollution environment, your biomarkers will change. And so we haven't looked for that, I don't think. Maybe you have. But there's no reason why you couldn't, so long as you have the instrumentation and you have the algorithms to mine that data. So this is just, I'll briefly just say that people have looked at pesticide exposure, and it does correlate with areas where pesticide use is higher. I'll let you dive into the papers because I don't want to go too much over time, or I don't want to go over time at all. And alcohol is, you've been told, is just another one of the very sort of metaphorically low-hanging fruit, in the sense that we use it a lot. Nicotine could have been something, or is something that people do quite ubiquitously, and there are biomarkers for that. So you can get these metrics for human behavior, but the metrics can go as deep as you want to look. And that's where this study went. So in Australia, they had a census, and they also have quite a lot of expertise in the area of biomarkers and wastewater epidemiology. And so what they did is they did one of the first sort of deep dives on just like what can we find, and how does it correlate with the things that we know about the society in Australia. And without really diving into the weeds on this one, they have found relationships between the presence of some chemicals in the wastewater and the population from which it came. And this is probably almost a proof of principle that it can be done, and I think the biomarkers that are present that aren't being mined, because these are largely prescription drugs, which will correlate, and you could have done it on paper if you had all the data for prescribing and compliance. But what you can do in principle for humanity is that you could understand this at a much greater resolution and not rely on prescription data. You can actually use biomarkers. Right, so this is another comfort zone of mine. So I was very active during the pandemic looking at sewage and trying to find SARS, and there was an academic consortium nationally, and Imperial was part of it. And we basically spent our time trying to develop the methods, trying to understand what mattered, what didn't matter, as far as handling the samples, collecting the samples, analyzing the samples, how do you analyze it, and how do you report it, how do you get Public Health England, which then became UKHSA during the pandemic, how do you communicate this to them and the people on the ground and in a local sphere, because no one knew how to use this data. And so you knew it was great. You knew that basically you were looking at the shedding of a virus from a person who doesn't know that they're infected, they don't know yet that they're infected, or they know they're infected. And so those three groups are all shedding, whether they like it or not, into the sewage. And so what you have to do is go to the sewage works, take a sample. You know, there were lots of things that we had to compromise on during the pandemic, and one of the things was a grab sample, and taking a grab sample is not the greatest way to do it. You want, at the very least, you want an auto sampler, or you want something even better than that, which is something that we spent a lot of time thinking about. But I won't talk about that right now, but effectively, just to show you what the pipeline would look like, there would be someone at the sewage works whose job it is to basically throw a bucket into the sewage, pour it into a bottle, send it off to, via a courier to a central lab, which the Environment Agency was primarily that lab, and then it would go through a pipeline of preparation, and qPCR was effectively the output of that. And you would basically have a value per day, and that might be every day, it might be 4 days a week. It varied depending on where it was in the world. And so this is just, this is what it looked like for a bit. So right about January, well, just after the Christmas period of 2020, going into 2021, they expanded the network from sewage works to manhole covers as well. And so I think this is the London manhole cover suite at one point. And so each manhole cover is on the left-hand side, and the date of when the sample was taken, and then the color coding, the darker it is, the worse it was. And you could see that you have some dark colors right next to really light colors, and that's because it's a grab sample. And this is why grab samples aren't good, because if you collect it, when someone flushes the toilet who's sick, you're going to get the toilet, you're going to get a very positive sample. And if you flush it in this, randomly, stochastic kind of period where, someone took a bath and you have a lot of bath water going down, so it's pretty clean looking. And so you have these sort of problems with the data. And so it was a small nightmare to figure out how to best use this kind of data. In a much smaller scale, we did some work at schools, so primary schools, secondary schools, and basically tested out the concept of, can you do near source sampling for wastewater, looking for COVID. This was done right at what I would call the beginning of when we were doing genuine wastewater surveillance in October of 2020 is when it sort of started, and then it basically continued for a year. And at the start, we were we were optimistic that fundamentally we could deliver data, but whether we'd find anything or not is actually mathematically quite questionable. So it was quite a roll of the dice to even go down this road to generate data, because if you think about it, there's 1,000 people in a school, you have an infection rate of maybe 2%. So you're already talking about, there's only 20-ish people walking around who are infected, whether they know it or not, asymptomatic. And then of those 20 people, maybe 10 to 20% might use the toilet. So now you're down to, almost no one is going to contribute a sample. And then you have an auto sampler set up outside of the school, taking a sample every whatever time period it is. It could be 1 minute on, 1 minute off, could be every 5 minutes, could be every 15 minutes, whatever the schedule is, because that's what auto samplers do, they run on a schedule. It's off for a period. So you're only capturing half of the data that's available. And so if there's only one person who's infected who's going to use the toilet, and you're only collecting half of the data, what's the chance of you having the machine on at the time that the water, his or her toilet flush goes by you? it's pretty low odds. But by the time Christmas came around, virtually every sample was positive. So one of those, or multiple one of those assumptions I just walked you through is really wrong. And it might be more than one. And so, you could start looking at the evidence right now of, people who with COVID have gastrointestinal upset. Now, if you were a normal kid, you might want to not take a poo at school, so you'd hold it in until you get home. But if you have COVID, whether you know it or not, you might not be able to hold it in. And so your contribution rate to this might be near 100%. And so that might be one assumption. The other one might be that virtually every kid was basically walking around with COVID, whether they knew it or not, by Christmas time. And so there's a combination of assumptions that play into a data set like this. This is an absolutely fascinating area. So you dive even further into wastewater surveillance, and you ask the question, could I find one infected person in a city? And the answer is sort of yes. So the study presented here was able to answer the question over a 6-month period because the person never stopped shedding the virus. And they never found who that person was. But it was really handy that they never stopped shedding the virus because they started at a wastewater treatment plant, which is on the left-hand side. They then found which of the branches of the wastewater, the network, was the one generating this COVID signal. Now, the reason that they were able to track this one person is because the word is, what is the word? Cryptic, yeah. They're looking for cryptic mutations. So they found a COVID virus that had mutations that have never been seen anywhere else on Earth. And they had so many of these mutations that you couldn't possibly miss it if you were looking for it. Now, everyone was not looking for it, and so everyone did miss it, but this person actually spent a lot of time looking for cryptic lineages. And so they knew exactly what they were looking for. And then they tracked it up and up and up and up, and eventually got to a building where basically one of these 30 people has COVID, but no one fessed up to it, so it remains a mystery as to who it was. And so that journey, so that was in Wisconsin, and it's still going on, and this is just a Twitter thread that if you find this story interesting, it's an active one right now, and they're still trying to figure out this Ohio cryptic lineage, which is floating around. And the human health implications of having cryptic lineages are, well, A, we didn't know they existed. You might have sort of theoretically assumed that they could have, but people are now measuring them. How frequent is it to get a cryptic lineage, and how medically important is it that they exist? Because it's a lot of diversity sitting out there inside of these cryptic lineages, but if they don't spread, then you could make the argument they're just academically interesting. Right, I'll speed through the rest, I suspect, but this is a really important part of what comes next. So we've built all this infrastructure for COVID, and it would be an utter shame if we didn't do anything with it. But how do you justify doing something with it to people who have a limited amount of money to spend? And so the phrase that people have started to use is the always-on approach, which effectively generates a rationale for why you would keep a wastewater surveillance network up and running in an inter-pandemic period. And so maybe we shouldn't spend a lot of time diving into each one of these, but the argument is that you need to make the case to politicians that the money spent on keeping this live during the inter-pandemic period, A, generates value to public health during this period, so you're generating useful, actionable data, and by having the expertise and people like me and people like Leon and his, the team, and everyone else who's globally who has invested time into this topic, if it's not supported, we will go to wherever the next project is, and if it ain't this, we're going to be somewhere else. And so the expertise will be gone, and we'll just have to start from reading the papers from whatever year it was to learn how they did it and how would we do it again. Would we do it any differently? Probably not, because no one funded the research. So this is an opportunity to, there are a lot of things that we did on the fly, on a shoestring. It wasn't good enough, but it got the job done-ish. But we know we could do it much, much better. And so if an always-on approach was supported, we would be developing all of that know-how. And to that end, there are people already talking about establishing this always-on, and the most logical place for it to start is in international travel. So there have been studies already that looked at airplanes and the toilets in airplanes to see how much is coming into each country. And despite the fact that, for example, China had a requirement that you couldn't get on a plane leaving China having not taken a test and testing negative, every flight out of China had COVID in its tanks. Now, that was probably always the case during the entire pandemic. It's just really hitting home the fact that there's asymptomatic carriage or pre-symptomatic shedding that is going on all the time. And it sort of talks to the point of why we underestimated the schools project. Another great example of how we can use the always-on concept is that monkeypox, there's always something coming around, unfortunately. And monkeypox is a great example of one that you would not have predicted should show up in wastewater. In fact, SARS shouldn't have been there. And influenza shouldn't be there, but yet everything is there. And so this is the really interesting part about wastewater epidemiology, is that it challenges the clinician who says, well, it's a respiratory virus, why would it show up in your stool? And it's like, well, I can't tell you why, maybe, but I can tell you that it is definitely there. And you can't argue against the fact that it's there because, there's millions of pounds that have been spent on setting up networks to find it. And monkeypox is another great example. I just thought I'd highlight a few imperial studies. And this is one of the more recent ones. It's not just imperial, but you were privy to some samples that were taken during the pandemic after having the routine monitoring of polio, which is done in London and Glasgow once a month, just to look to see is there anything unusual going on. And one might have to assume that nothing interesting has been going on for a while because no one heard anything about a polio outbreak. But funny thing is, once you start collecting the samples, people start finding stuff. And so they started to find polio that shouldn't have been there. And so they went back to the samples that had already been collected. And those samples turned up positive. And much like the Israeli study, they started looking at mutations and showed that there was human-to-human transmission, potentially. And so go in and vaccinate. So, in conclusion, in conclusion, wastewater is a fantastic resource, and we absolutely must be supporting the always-on approach because we'll be back to where we were 3 years ago if we don't, and that would be a shame. So thank you very much for your attention.
Chemicals for infectious disease, fascinating resource. Thank you, Andrew. I'll allow one question quickly before lunch. Digging in. Let's have it here.
Sorry if this is a simplistic question, but we know that with infectious diseases you have like super shedders. And
Can you just use the microphone, please? Yeah.
We know that with lots of infectious diseases, there's variation between individuals and how much they shed, length of time, and the intensity of shed. How do you account for that? And is it the same with pharmacokinetics as well? Is there a lot of variation in individuals and how they metabolize chemicals?
So that's a lot of questions, but
Sorry.
That's all right. Yes, humans will be different, and some will be absolute massive shedders. So the Ohio study, he calculated an unbelievable amount of shedding that was going on, to the extent that he thought that the entire gut lining of this person, or intestinal lining, had to have been shed like on a daily basis. to achieve the amount of viral load that was coming out into the sewage. Now that's a really unusual example, we think. But the thing is that this is really early days, so we can't really say anything conclusively other than there's a lot of variation. It depends on when you were infected as to when the shedding has maximized. So it's usually right about when you're starting to realize, I'm not feeling well. That's probably your highest shedding day. And then it, typically will tail off, but the tail could be 20 days long. It could be 40, it could, like this person was going on for 6 months plus. So now, how do you account for it from an epidemiologist's point of view? How does someone like that use this data? Well, that's a good question. And so there are tricks to doing this. So you could do it, the hard grunt way, mathematically, or you could do it, it's going down, it's going up, it's staying the same. So you look at it, each location over time, and you compare it to its predecessor day, or you can do what we did with the really awful manhole cover data, which was really noisy, is that you actually just average 3 days or average 5 days, so it's a five-day rolling average. So there are ways to sort of smooth out the data, and this room could probably come up with 100 different ways, and all of them should be considered. So I don't think we've sorted it, and that's why I think it's also really valuable to have an always-on approach, because no one will be thinking about it if there's no need for it right now. So if we all just sort of say we're done, wait for the next pandemic, no one's going to be worrying about this problem. But it is a problem.
Okay, thank you. We'll stop there. Fascinated by the idea of the air traffic sewage tanks on the airplanes. That'd be interesting, tracking what's on those. So a round of applause, please, for Andrew.